Okay. Hello and welcome to our PolarConnect event. I'm Janet Warburton. I'll be hosting this event today along with Judy Fonstock. We both work for the Arctic Research Consortium of the United States. I'm based in Anchorage, Alaska, and Judy is in New Hampshire. Today's presentation, we're excited that we have a teacher in the field in the Arctic working with researchers. Rebecca Siegel will be talking about the harmful algal blooms in Arctic waters, a project that she has been associated with maybe less than a year. It's been quite the whirlwind adventure for her. She's out at sea. The whole team is out at sea right now. So this broadcast is coming from the Arctic Alaska and off the coast of Arctic Alaska. She will introduce her team members here in a little bit, but before we get started, there are a few things about these PolarConnect events I wanted to share with you. The first one is it is being recorded. After the event is done, Judy will send out the recording to everybody that's registered. And I forgot that I'm in charge of my own slides. Here we go. In the meantime, we hope that as participants, you'll introduce yourself in the chat box. Let us know if you where you're joining us from and if you have any students or others in the room with you. A little bit about Polar Treks and how it fits with the Arctic Research Consortium of the United States, or ARCUS. We've been hosting this program since 2004, and it is an opportunity where teachers are placed with field researchers, and they go out to locations throughout the Arctic and Antarctica. I see a few alumni that both researchers and teachers have joined us today. We are excited because the last couple of years due to COVID, we haven't had any researchers or teachers with researchers. Oh, another little handy thing is if you have a microphone, we hope that you will mute yourself while the presentation is going on, and so that way it can focus just on the research team and the presentation. At the end, we will have a time for questions. You can also post them in the chat box and we'll relay them as we go along. But at the end, you can open up your mics and ask your questions. Okay, I think that's it. I'm gonna turn this over to the team. They were muted, so now they need to unmute themselves if they're able to. There you go. And good morning. Please just let us know how it's going. And when we go through slides, just say next slide for me so I know how to navigate. All right.
Hi, everyone. I'm Rebecca Siegel. I'm a teacher from Brevik Mission, Alaska. I teach at the Brevik School, and I'm really excited to be here on the Norseman II. And I like she and her pronouns, and I'll just let everyone introduce themselves.
Hi, everyone. My name is Bob Pickard, he, him. I'm a physical oceanographer from the WHOI Oceanographic Institution. That means I study where the water goes, how it moves, what the properties are. So I'm not a biologist, I'm a physicist. I'm also one of the co-PIs, along with a biologist from my institution, who's actually not on the ship this time. Yes, I'm also the chief scientist. So I'm sort of keeping track of what we're doing and, what our future plans are as we progress through the cruise.
Hi, I'm Evie Fashon, she, her. I'm a student at Woods Hole Oceanographic Institution. I work with Don Anderson, who is the co-PI on this project, and he is not on the ship, but our lab studies harmful algal blooms. We've been working up here in this region since 2018, and we're really excited to be back this year and taking a look at what is in the water. My name is Kate Hubbard. I'm a research scientist at the Florida Fish and Wildlife Conservation Commission, and I'm working on harmful algae. I'm also a member of Don Anderson's lab at WHOI, and I'm excited to be here collecting DNA samples, toxin samples, adding some different dimensions to our studies. Nice to see everyone.
Hi, I'm Jim Wells. I am the engineer on the Norseman II, and I'm here to keep everybody safe and operations working for them so they can get their tests done. Glad you guys are all here.
All right, so that's our team. I'm going to turn our video off now because just to cut down on the bandwidth. So you'll hear us, but I guess you won't see us. All right. And we could go to the next slide.
Rebecca, the
All right, so
slide is up. I was just going to relay to you, Rebecca, this is Janet, that there is going to be a delay on your end, but rest assured that we are moving through the slides as you requested.
So this is the rest of the team. There's 24 people in all on the Norseman II, which is about 117 feet, so it's pretty cozy. But on this team, we have one person who has 1.7 million followers on TikTok. We have somebody who ran 96 kilometers in 13 hours, and we have somebody who's going to retire at the end of the month. So pretty good group. All right, next.
Okay, Rebecca, you can go ahead. The next slide is
Okay,
up.
and here's our captain, Casey Coates. He is up in the wheelhouse right now, so he can't say hi, but here's a picture of him at his station with his different displays for radar and the depth and our course. All right, next. Okay. And so where are we? We're on the Norseman II, which is it's a 117-foot boat. It used to be a crab boat, but it was bought out of the fishing fleet and converted to a research vessel. And we are, you can see on the right, a map of our track, and we are right in that red circle in the Chukchi Sea, quite close to the border with Russian waters. We set sail from Nome back on July 19th. And headed down towards the mouth of the Yukon River. And then we've been following this line ever since. You can see the blue dotted lines are where we're taking samples. So every blue dot is a place where the ship stopped and we collected some data and samples. And then the black lines are the transits in between the transect lines where we're sampling. Okay, next.
Okay, Rebecca, it's up.
All right, so we did see Russia. We've seen it quite a few times. As I said, right now we're very close to Russia, but this picture was from a clear day when we were off of St. Lawrence Island, and we could see an area called Providenia that you can see circled on the map in the upper right corner. So it's been pretty cool to be so close to the border. At one point, my phone changed itself to a Russian time zone for a few days. So that's quite interesting. Next. All right, so we are here studying phytoplankton, and phytoplankton are small organisms that float with the current. They can't swim against the current, and they use sunlight to make their energy, so they do photosynthesis. And so a byproduct of photosynthesis is oxygen, and it turns out that phytoplankton produce more than half of the oxygen in the atmosphere, so they're pretty important. And we have one instrument on board called the Imaging Flow Cytobot that's able to sample the water as we're steaming along and then take photographs of any plankton in the water. So this is one set of images that it took, and you can actually see those big dark plankton are Alexandrium, which are one of the main ones we're studying that you'll hear more about in a minute. Next. All right, so in the right conditions, phytoplankton can bloom or multiply rapidly. And usually that's not harmful, but sometimes you get harmful algal blooms. So that's when some species bloom, they can be hazardous to humans and marine life. And this can be in a variety of different ways. Some are toxin producers, which are what we are focusing on this cruise. One toxin producing plankton causes paralytic shellfish poisoning, and another one causes amnesic shellfish poisoning. All right, next. So the first plankton that we're focusing on this cruise is Alexandrium catenella. This is a phytoplankton that produces saxitoxin, which is a neurotoxin that can cause paralytic shellfish poisoning. And if someone had paralytic shellfish poisoning, their symptoms would include paralysis, tingling, slurred speech, vomiting, and then because of the paralysis, your lungs might not work. So in severe cases, they need to be on lung support until the toxins can pass out of the body. And Alexandrium is pretty tricky because it has a very unique life cycle. If you look on the left, you can see a little diagram. So it has the main stage, which is the vegetative cells, which are the plankton floating in the water. And then those can bloom, but then they also can produce a resting cyst, which is a little egg kind of thing that is down in the seafloor. And then when conditions are right, those cysts can bloom back into the vegetative plankton in the water column. And the cysts can last for years and years, waiting for good conditions. So that's a pretty important part of their life cycle that we'll be looking at. All right, next. All right. So let's take a look at those cysts again. So this is a map from data from the years of 2018 and 2019 showing where the cysts are in the Bering and Chukchi Sea. And you can see that there's a big cyst bed in this area called Ledyard Bay, and then another one in the Beaufort Sea. And so what we find this year might look similar, it might look different because it is a few years later. But the main information is that there's a big cyst bed in the Arctic, and with climate change, the waters are warming, so that increases the ability of the cysts to germinate and bloom. And so we're here to study the cyst bed, to look at the Hab cells in the water, and work on understanding the ecology behind the blooms. We're not a monitoring expedition. Instead, our focus is to establish the basic ecology, to understand how the Alexandrium work in the ecosystem, and that can help us with modeling and predicting. And knowing the place of Alexandrium in the ecosystem can help us to move forward. Okay. Next. So our other big Hab plankton that we're studying on this cruise is Pseudonychia. And this is a diatom. There's at least 8 different species in the Bering and Chukchi Seas, and some of them produce toxins and some don't. The toxin that they produce is called domoic acid, and it can cause amnesic shellfish poisoning. Some symptoms are short-term memory loss, vomiting, erratic behavior, and seizures. And we're here to figure out which species are out there, how toxic they are, and then to understand their ecology. You can see from this picture from the IFCB that they often form these little chains. All right, next. So phytoplankton are really important because they form the base of the Arctic Ocean's food web. And this is how the toxins make their way from these microscopic plankton into a person. So we're here also trying to understand how the toxins travel through the food web. And there's 2 main sort of food chains that we're interested in for how the toxins can reach humans. So the first one is that the phytoplankton feed the clams and worms down on the seafloor, and those in turn are food for the walruses. And walruses eat about 10,000 clams and worms a day, so they're consuming a great deal, and the toxin can accumulate in walruses possibly. And then humans eat both clams, so they could get the toxin straight from clams, and then humans also will eat walruses. Walruses are a really important food source in this region. So that's one of the food chains we're looking at. And then the other one we're looking at is on the next slide. So another really important food source in this region are bowhead whales. That's important subsistence food. And again, the phytoplankton are the base of the food chain. So they feed zooplankton. Then the bowhead whale will feed on the zooplankton. And then humans will eat bowhead whales. So we're also trying to understand how the toxins can affect the bowheads. All right, next. All right, so how do we study this? How do we study how the toxins travel through the food web? So one of our main tools is this instrument here, which is called a Smith-McIntyre grab. And you can see it has these two rounded jaws, and it's on a crane. So it travels down to the seafloor. When it hits the bottom, it triggers and the jaws snap shut, giving us a sample of the mud on the seafloor. And next. And then we haul it back up to the ship and open it up. And the first thing that happens is we take a sample of the mud from the seafloor, and that will be used to count any cysts that are present to create a new cyst map showing where the cysts are in 2022. And then we collect the mud in a cooler that you can see down below the this wooden apparatus. And then if you go next. The next step is to use a hose to rinse all the mud out of through a colander and leaving behind any clams or worms or other organisms that are left. And then one of the scientists on board will take those organisms back to his lab in Washington State, and he will analyze them for any toxins that are present. So let's take a look at some of those organisms that we're finding on the seafloor. Next slide. All right, so organisms that live on the seafloor are called benthic organisms. And here on the left, you can see Patrick preparing them for transport back to Seattle. And I'll take a moment to show some of the ones we find. So on the upper right, you have some gray clams. These are macomas. They're find a lot of those. And then below we have a Serepes clam, and then we have quite a few amphipods, little shrimps that we find. And then next slide, we'll show some worms that we find. All right, so then we also find a lot of worms. On the left-hand side, you can see a shrimp. We only found one of those big shrimp, so we're pretty excited about that. Some little macoma clams. And then the worms on the left are called Scalabrigum inflatum. And then on the right-hand side, you can see an ice cream cone worm, or a pectinaria is the Latin name. And they form, on the far right, you can see the shell they form out of little grains of sand that are fused together. And then if you take it out of its shell, you see this red worm with little golden bristles. Okay. Next, please. So we're also studying the oceanography. To understand the plankton, we need to understand the water they live in. They can't swim away to find a spot, a better spot. So if the water they're in gives them the right conditions to bloom, they're going to bloom. And if it doesn't give them the right conditions, they won't bloom. So we're studying the different water masses to learn about the conditions the plankton are living in. And we do that with this big instrument here called a CTD, that stands for conductivity, temperature, and depth. And conductivity is how well the electricity can travel through the seawater, and that's a way to measure salinity. So what we have here is this big round frame that's called a rosette. At the top, you see some dark tubes, vertical tubes. Those are called Niskin bottles, and we use those to sample the seawater at different depths. And then below that is kind of a mass of wires and sensors, and that is the CTD. So we send that down to towards the seafloor and then land it back on the ship, and it creates quite a lot of data. We'll take a look at some of that on the next slide. So here's a CTD plot. This is the data that the CTD sends back even as it's descending down to the seafloor. And this one shows depth versus temperature and salinity. So let's take a little look. On the left, we have the depth measured in meters with the sea surface up at the top going down to the bottom. So the higher up is the shallower waters and the lower down is the deeper waters. And then in blue here, you'll see salinity. So you can see that there's this upper water mass that has a lower salinity, less salt in it. and then a transition layer, and then lower down there's a saltier water mass. And then in red is the temperature. So you can see that the surface water was warmer. There was a transition layer where it was cooling off, and then the lower water mass is colder. So we have one water mass on top that's less dense, that is warmer and less salty, and then a lower down mass that is denser, saltier, and colder. Okay. And then let's see what that means for the plankton on the next slide. Okay, so then we also have a sensor on the CTD that can measure how much chlorophyll is in the water. So remember, chlorophyll is a pigment used in photosynthesis. So the amount of chlorophyll can give us a sense of how many plankton are in the water. And what you see here, again, is the depth is on the left, and then this green line is the amount of chlorophyll in the water. And you can see that around 18 meters is a big spike in the amount of chlorophyll. This is called the chlorophyll max. So that's telling us that there is a density of plankton right at that level. And that might be because there's more nutrients there. There's some different reasons. Okay. So now let's take a look at some plankton on the next slide. So we capture plankton in two different ways. On the left side, you can see a plankton toe. This is a net with very fine mesh that can capture the tiny phytoplankton. So we'll lower that down about 30 meters into the water and then lift it up to catch any plankton in the water column. And then on the right, you can see the Niskin bottles that close at different depths to trap in any plankton that are there. So yesterday we did a plankton toe, and I took some of the plankton from that, put it under a microscope, and took some pictures so that you can see. So next slide. Okay, and Kate is our plankton expert, so she's going to explain what we're looking at.
So we have seen actually different types of plankton as we've moved through the survey, and this is showing mostly diatoms. Diatoms are unique in that they have silica cell walls. Silica is just glass. And so you can see in this picture a lot of different types. So there's a high diversity of diatoms. There's a few other things mixed in. Diatoms are also distinct because they're typically symmetrical. So you can see things that look like little hamburgers, and they form chains. And so this is just a huge concentration of them. And as we move through the next slides, you can just get a sense for the diversity of these special plankton. So here again, you can see some that are single cells. There's one right in the middle that is probably Coscinodiscus. We also have other chains, and those are the ones where you can see they're sort of piled up together, and they tend to bloom. And I don't know if Bob wants to comment on why we might be seeing diatoms blooming in this particular area.
Well, yeah, sure. So to bloom, you need the nutrients and the sunlight. And it turns out that one of the water masses that Rebecca was talking about was that cold water that's near the bottom. And that water tends to be higher in nutrients because it's actually gotten the nutrients from the sediments. And so that's what the phytoplankton are looking for. And hence they tend to kind of concentrate right at the top of that cold water where they still have enough sunlight, but they can still tap the nutrients. And that's why you get the blooms happening right there.
And Rebecca mentioned earlier that most phytoplankton are not toxic. And in these images, we're seeing a lot of non-toxic phytoplankton. Again, these are really important for forming the base of the food web.
Okay. The last thing I just wanted to talk about was life on a ship, because it's pretty different from life on land. And the main way that it's different is that operations continue around the clock. So everyone is divided into 12-hour shifts. For the scientists, there's one shift from midnight till noon, and the other one from noon until midnight. So pretty much at any time, there's somebody trying to sleep. So on the left, you can see our bunks, and so you can see there's 4 people in a little bunk room. So there's some different things to help give privacy and let some people come and go while other people are sleeping. You can see that there's a black curtain that you can pull around your bunk so that it's dark inside. And then every bunk has a little light inside so you can see if you're trying to keep it dark for other people. And then on the right, you can see the salon. This is the main sort of cabin of the ship where we spend most of our time. And the most interesting thing I think here is that you'll notice on the table, there's a blue covering, and that's a sort of a non-skid mat so that when you put your cup or your plate down, it stays in one place. And every surface on the ship has that on it so that things stay put. Any questions?
Good one. Thank you, Rebecca and team. Yeah, so now it's time for questions. Rebecca, you can turn back on your video. I'll stop sharing so that we can get straight to those questions. And if we need a slide or something, please, I can pull it back up. In the meantime, while people are thinking about it, I will relay a couple of questions that came up from to you while you were doing your presentation. So from Mark, how much has the water in the Chukchi Sea warmed in recent decades?
All right, so that's probably a question for the physical oceanographer. So one of the important aspects of these harmful algal blooms, as Rebecca was talking about, these cysts are sitting in the sediments, and they're going to remain dormant unless the water warms up enough for them to germinate. Okay, so traditionally up here in these northern waters, the waters have just been too cold. Okay, but everyone realizes that we're undergoing climate change, and it turns out that the waters that are feeding the Arctic through Bering Strait, the small little channel that goes from the Pacific to the Arctic, the water that's been coming through there has been warming up in recent decades. So one of the things we've done is we went back and looked at all the data that we could find from ships that have gone through the years, and we broke it into different 5-year segments. And we were able to show that over the last 20 years, the bottom waters, okay, have warmed up considerably. And now in the most recent 5-year period, in this area that we're looking at now, Ledger Bay, it's now warmer than 4 degrees Celsius, okay? Which is about, I don't know, almost 40 degrees. So yes, they warmed, and it looks like they're going to continue to warm. So that's a real issue up here.
Okay. Let's see. Oh, I was wondering how many different species of phytoplankton are you finding? Because you say there's a lot, but can you give us a sense of what a lot means?
Hundreds.
Hundreds.
It's hard to, but a lot.
Okay. Let's see. So we have from Aaron, what has been the biggest takeaway from your work with the team, Rebecca?
I think the biggest takeaway is how much everything is changing here, and from day to day, and then year to year, and then from climate change, and how they are able to respond to that and adjust plans and make the most of the time. I think that's just been really interesting to see how much change is happening.
Okay. Wendy and Dean are asking, what besides salt influences conductivity? And also, what is the temperature at 15 to 16 meters?
I said, what besides salt affects conductivity?
Oh, the temperature does as well, but primarily it's the salinity. The conductivity is influencing the salinity. So, and that's how we measure the salt content in the water. You know, in the olden days, we had those bottles, the Niskin bottles that Rebecca mentioned on our rosette. We used to have to capture the water, bring it into the lab, and you know, just measure it with what we call a salinometer. And that was really slow, and we got very few measurements. But now that we can do this electronically, it's really changed the game for us. So we can have these really high resolution continuous profiles everywhere we stop the ship. We can get a measure of the salinity from the top to the bottom. So it's really been a game changer for us. And there was another question about temperature, did I hear?
Yeah, what is the temperature at 15 to 16 meters?
Oh, well, that's highly variable. Okay, so the warmest water we've seen so far is probably about 50 degrees Fahrenheit, maybe a little bit more than that, and that tends to be up near the surface generally. However, in some areas we've seen water that warm down near the bottom. One aspect about where we are, the Chukchi Sea, is that it's a very shallow sea. It's only about 50 meters deep, about 150 feet. And so, the sun can really warm up the water. And then if you get any sort of wind blowing, it mixes up the water and can bring that warm water right down to the down to the bottom. So you can get pretty warm water down at the bottom, and we've seen that. On the other hand, we've seen water that's near the freezing point of seawater, okay, which is actually about 30 or 29 degrees Fahrenheit. So that's quite a contrast, and that's we're seeing near the bottom, and that's actually been a surprise. I did not expect to see water near the freezing point this late in the season, into August.
Okay. From Julie Brigham Gretty. Thanks, Julie, for writing this one. Did the center of Alexandria in the Chukchi move north out of the Bering Sea? So I might have to find the map,
That's a really good question. It's something that we are trying to address in some of these studies. We don't know now how long that big accumulation has been up in the Chukchi Sea, and we're trying to understand how connected those northern populations are to populations of Alexandrium in the Bering Sea, and even in the Gulf of Alaska, which has a really long history of algal blooms. And then also in the Gulf of Anadyr on the Russian side, we know they also have Alexandrium. So all of these different populations in the south have waters that travel through the Bering Strait and into the Chukchi Sea. And there are still some big questions there that we have to answer about that.
okay.
So
Yep,
let me
go
chime
ahead.
in. Evie just mentioned the Gulf of Anadyr and the Russian waters. So, and of course, Rebecca was talking about the fact that we can see Russia. So one of the issues that we're dealing with here is, and you can actually see it on the map in your screen, half of the Chukchi Sea is in Russian waters. And presently we are not able to access that half of the Chukchi Sea. So all of our research ships are confined to the right-hand side, right? So we get these beautiful pictures and all this data, and we can sort of make sense of what we see in our waters. But, for instance, with this study, we're thinking that a lot of the waters that are feeding Bering Strait are coming from the Russian side, but we're not sure. And it would be really important if we could get in there and measure that. And the other thing is, once you're in the Arctic, once you're through Bering Strait, a lot of the water goes to the left, goes towards the west into the Russian side, where you see the blank, right? So when you look at a picture like this and you see this big seabed, this bright red color right near Ledyard Bay, you say, "Oh my gosh, that's a big area." And in fact, I think it's the biggest area in the world ocean that's been measured to date that's seen, that's characterized by this high seabed. But I would bet that it's even greater on the Russian side. But unfortunately, it's frustrating we can't get over there and measure that. So that's one of the issues that we constantly have to deal with, the fact that we're only measuring half of the Chukchi Sea and half of the Northern Bering Sea.
All right, thank you. Okay, let's go to Stacy. Stacy had a comment for you, Rebecca, and a question. Rebecca, your expedition sounds similar to my experience on the RV Atlantis with 12-hour shifts and whatnot. How's the food? On such a small ship, what do you do in your downtime?
Oh, those are great questions. The food is fantastic. This morning there was cereal and oatmeal, and I think there was eggs and bacon and potatoes and toast. So there's lots of choices at all the meals in our cook is kit, and he has a very small galley, and he has things stored all over the place. There's a freezer that has his food and also samples, kind of sharing space, and sort of any storage area, there's like cans of tomatoes and whatnot. So he does a great job. And then downtime, see, I like to bird watch, so I'll go out and bird watch. We have an exercise bike, so lots of people are taking advantage of that. And you can play cards. We're having a cribbage tournament right now. Read. There's a TV. We've watched a couple movies, so we keep pretty entertained. It's been a lot of fun.
And none of us are birds, so Rebecca has been teaching us all about the birds that we've been seeing on the cruise. It's been really nice to have her expertise.
Yeah, have you seen any unique bird species, Rebecca? Or like?
Yeah, I was very excited because I saw a thick-billed murre and I'd never seen one before. And then every day there's been short-tailed shearwaters and black-leg kittiwakes following the boat. And we've seen a couple puffins.
Cool. Well, that's nice. All right, from Ken, we have a follow-up for Bob. How has the circulation in the region changed in the years? In recent years, sorry.
That's a kind of a difficult question to answer, how it's changing through the years. I guess probably that one of the main differences is that the amount of water that's coming from the Pacific into the Chukchi Sea through Bering Strait, through that narrow little passage, that's been increasing through the years. And we know that because we have what we call moorings that we stick out in the water year round, and they can measure how much water is going by the mooring. So it's increased about 30% over the last, I'd say 15 years or so. And it's so there's more water, and it's also becoming warmer and fresher. So those changes are going to be felt further, downstream into the Chukchi Sea, and we're still trying to figure out what that means, how the circulation is changing because of the fact that there's more water coming through the strait. And that's something that we'll learn with more cruises, not just our cruises, but cruises from the international community. We put all the data together and we try to figure all that out. But I will mention one other thing though, that's talking about long-term changes. But one of the most striking things about the circulation is how quickly it can change, okay? So today we're sitting here and it just so happens we're having our video chat while the wind is blowing and we can't do any science, okay? Because the sea is too rough for us to do our science right now. And that's, again, it's because of the wind. And so when you change the wind direction or you increase the wind, it can really change the circulation quite dramatically in a short amount of time. So one of the things that we're studying are these currents that are going towards the north, okay? And if the wind blows hard enough, the current that's right next to the coast of Alaska can completely turn around and come in the opposite direction. And that can happen in a matter of hours. It's just remarkable how sensitive the circulation is to these changes in wind. So you got to sort all that out as we're trying to do all the biology. We're trying to keep track of all the changes that's happening with the circulation.
All right, great responses and great questions. Here's one from Elaine. Do you see less phytoplankton during the times of the year when there is less sun in northern latitudes?
Yeah, they're pretty much going to be able to respond to light and nutrients wherever that is. And one of the things that can happen as sunlight gets a little bit scarce in this area, there's also typically sea ice that forms. And so some of the plankton that we work on, the phytoplankton, can actually live in the sea ice. And so they're able to kind of eke out a living until things open up again and things warm up. But I think with the diversity of phytoplankton that are out there, they are remarkable in terms of being able to occupy every niche, kind of like where we have food stored in every possible place on the ship. The phytoplankton are really successful at trying to take advantage of all of those little pockets in the ocean that they can live in. But typically, we might see that things get a little bit less, there might be less productivity during the winter just because of that light limitation.
All right, thank you.
So can I just add to what Kate just said? So she talked about the impact of the ice. And so, one of the huge changes that's happened over the last couple of decades is the fact that we have less pack ice, right? So in the olden days, and when I say olden days, maybe 20 years ago, 30 years ago, so not that olden, we wouldn't be able to come up here on a ship like this because the Norseman II does not have an ice strengthened hull. and you would need an icebreaker to come up here. So now we're up here with this ship that, it's an open water vessel, and we're not seeing any sea ice, right? And so of course that's allowing the sunlight to come in. So not only do we have a longer stretch of time when there's sunlight that's able to get into the water because we don't have the ice, but even when we do have the ice, it's thinner than it used to be. And that allows more light to get through that thin ice to get into the water column. So the lack of ice and the thinner ice all means more light is getting into the water, which is going to impact the biology.
Yeah, great. Thanks for bringing that up. Nina is asking, the Chukchi Sea is relatively shallow. Does the winter ice keels scrape the bottom and release the cysts?
Cysts. That's a great question. So yes, the Chukchi Sea is very shallow, and so what happens in the winter time is that the cold air starts to cool off the water, okay, and that starts to make the water a little bit dense on the top. And if it gets dense enough, then it can actually sink right down to the bottom. And then when the ice forms, what we call it rejects the salt, okay? So when the ice forms, it leaves some of the salt in the water, and that also makes it dense. So the fact that it's cold, the fact that it's salty, means that it can sink. And since the Chukchi Sea is so shallow, it can sink right down to the bottom very easily, all right? And down on the bottom is where we have a lot of those nutrients in the sediments, right? And by the water coming down and hitting the bottom, it can just pull those nutrients up into the water. And that's why the cold water tends to have the higher nutrients. So the fact that the Chukchi Sea is shallow is a huge part, really important part of this whole system.
And then as far as the Alexandrium cysts go, they are generally only active in the spring and in the summertime. So we're curious to know what effect ice might have on them, but they're not germinating or blooming in the wintertime. They're just staying in their cyst form.
Okay, and you might have answered this next question to some extent, but I'll repeat it just in case there's something you want to add. Mark is asking, to what extent do you think the loss of sea ice influences the productivity of phytoplankton? And you kind of touched on this, so.
Well, yeah, so like I said, it means more sunlight getting into the water. And I also mentioned the fact that if the ice is thinner, you can also get light through the water column. So one of the huge discoveries that we've made just in the last few years, last 10 years, is that we're starting to get what we call under ice phytoplankton blooms. That again, didn't used to happen back when the ice was several meters thick. But now the ice is thin enough that it's allowing the whole system to get cranking earlier in the season, because they're getting light into the water column earlier than you used to. And another aspect of that is that when you melt the sea ice in the spring, you start to form these what we call melt ponds. You get these little ponds of water on top of the ice, and those are like little skylights, and you can get significantly more light coming through these little ponds into the water below. And that's really fueling these under ice blooms. So ice is a huge part of the story, and the fact that the ice is changing is also a huge part of the story.
I don't know, Evie was able to grab some ice on surveys in 2018, and we were able to do genetic analysis on that, and we did find some pseudomonas species that were in the ice. So it's something that I'm not sure if we'll find any ice on this trip, but it's certainly an interesting part of trying to think about the harmful algal blooms and what seasons they may be able to occur, and then also as things are changing, what might happen in the future.
I'm going to kind of jump to one question because while you are all talking, Ken is asking, are there any microplastics or are microplastics an issue in your study area?
I know that they have been found up here. We had in 2020, Nicole was on the boat. She was sampling for microplastics, and I haven't heard what the results of that are yet. None of us are microplastic experts, unfortunately.
Okay. Stacy is asking, yeah, are you using other types of data collection, satellite float, along with the ship data, CTDs, to find the phytoplankton blooms?
So on this cruise, we are using satellite data to look at the ice and where the ice is, which is helping us plan our track and make sure that we're making the most of our sampling time up here. Alexandrium can cause red water if they're in high enough densities, but they have to be really, really dense to be visible by satellite, at least with the technology that we use now. So even though the densities of cells that we're seeing up here can be enough to cause toxicity, they're not dense enough to be able to see from space. So it's not a useful tool yet, but it could be in the future.
Okay, that kind of leads into Amanda's question about how are the transect locations determined?
Okay, so yeah, they're determined partly because based on what we saw, Evie was talking about, you know, the seed bed that we previously mapped out. So we wanted to target that area. So we did some of our transects in this region that we're still in the Ledger Bay region. But we also, you know, wanted to cover the whole system from the northern Bering, you know, to see what's coming into Bering Strait, all the way through the Chukchi, and then sort of what's exiting the Chukchi Sea, very at the top of Alaska there, the northernmost part of Alaska. So it's sort of just, you know, we want to do enough lines that we get good coverage, so that gives us a good, you know, area to cover. But at the same time, Rebecca talked about all the dots, all the places that we're stopping. We want to stop often enough that we get a really high resolution view. Because if we don't stop often enough, we're going to miss things, right? We're going to miss flow. We're going to miss, you know, harmful algal cells that are coming through. So you have this combination of trying to cover as much area as possible, but also covering it at high enough resolution. So we're constantly juggling those two things, plus the fact that we only have 28 days out here to try to do, you know, maximize the ability to see what's going on.
Do you want to talk about the DDO lines? Just that we're visiting a lot of the same stations that other researchers are visiting, and that helps provide more and more data points from the same location, so we get a better sense of how things are changing over time.
All right. A question for Rebecca from Emma. What are you most excited to share with your students?
Really excited with them to share how the plankton communities are related to the sea conditions and the properties of the water. And I'm hoping when we get back to Brevik and we have the lagoon and then we have the ocean side by side, so we have these two very different water bodies with very different water properties that are, you know, 3-minute walk from the school. So I'm hoping we can collect some data about those and they can make some discoveries of their own. And I'm hoping they can get a sense of the excitement of doing science and getting, you know, going out when you don't know the answer to the question.
All right. Nina was wondering, have any of the indigenous populations or locals encountered more incidents of the poisoning that you mentioned?
So up here in the Chukchi Sea, there really isn't a history of like local communities experiencing paralytic shellfish poisoning or amnesic shellfish poisoning. Down in the Gulf of Alaska, there's a really long history of PSP events and even fatalities. And I think on Alaska a couple of years ago, there was a really unfortunate paralytic shellfish poisoning fatality. So in this region, this is a newer thing that we're concerned about, and we're really lucky that it's not something that's seriously harming people yet, as far as we know. That's part of why it's important to get a handle on what's going on with these blooms now, so that we can put preventative measures in place.
Okay. A few more questions here that came up. Kay Conger wants to know, has there been any cooperative projects with Russian scientists? You talk about that data gap. Has there been anything going on there?
Yeah, so our group actually received funding last year to collaborate with some Russian scientists who were working just on the other side of the strait and to share data and even have them come over here to Woods Hole and work in our labs and do some in-person collaboration. Unfortunately, given the current geopolitical situation, that effort's on hold for the moment. So we're kind of waiting to see if we can keep that moving. It's really important to be able to share that data with them. So it's something we would like to see move forward, but we just have to be patient for now.
Yeah. And Stacy is wondering, have you seen any of the animals that were at the top of your food chain? Whales, walruses, etc.
We've definitely seen some whales. I know we saw orcas and I think gray whales. We've not seen any walruses. They tend to be where the ice is, so we can't get too near to them.
Okay. And Sandy wants to know what sea state will prevent you from being able to work, and how many days has this happened?
Okay, that's $1,000 question, right? So we're on a fairly small vessel, all right? It's 115 feet. And so it is sensitive to the sea state. And today we're not working, and the seas are about 3 to 4 feet. So that's about our cutoff here, 3 to 4 feet in terms of being able to work or not. And what's a little bit frustrating, it's not just dependent on what the winds are doing, because the winds are not all that high right now, even though we can't work. And the reason why is because we're getting remote swell from other places that come in, and they keep us from working. So we started our cruise on a harsh note, right? It was blowing so hard that we had to leave the dock at Nome and experience weather right up front. And I think a good portion of the science party was kind of under the weather at that point. So we've lost probably 3 days worth of time now, and it's continuing as we speak. And again, it's been a little challenging because if you're on the larger vessel, then you're able to work in these kind of conditions. And this is a smaller vessel. Now, I'll hasten to say one of the advantages of being on a smaller vessel is we can get very close to shore. In some cases, we've gotten as shallow as 12 meters. Okay, so about 30 feet or so. And that's a lot shallower than you can get if you're on one of these really big vessels, like a big icebreaker. So there are advantages of being on a smaller vessel, but it is a challenge due to the weather.
Okay, and I think that probably answers Stacy's other question was, has anyone gotten seasick? So based on that response, under the weather, I take it a few people did not feel very good during some of those storms. All right, I think that sums up most of the questions. If I missed somebody's, I'm sorry about that. Please feel free to repost them, or you can also post them in respond to Rebecca's journals on the Polar Trek website and repost your question there, and she can get it answered by the team. Oh, one just came up very quickly from Emma. What is the vessel size? You did mention that it was smaller, and I think actually you said it earlier, but what is the size of the vessel?
It's 125 feet. And just contrast that to the big icebreaker that's owned by the Coast Guard. That's 450 feet. Okay, so that's quite a difference.
Exactly. Yeah, so we want to wrap this up very quickly. Thanks again, team, for sharing your science and also sharing just being able to see you and hear what you're doing. Very interesting and well presented presentation. Before we wrap up and everybody takes off, we just wanted to let you know we do have other educators like Rebecca that are heading into the field. The next group to head off into the field is actually down to the other polar region, to Antarctica. We have 3 teachers that will be going down there this year. So please stay tuned and what is happening with Polar Trek. Here's some things you can, where you can find us and what is happening. And we also want to thank you all for joining, and especially to the National Science Foundation and Office of Polar Programs for hosting and funding Polar Trek program. And with that, again, like I said at the beginning, we will have this recording and it will go out to everybody that registered and will be available on the website. Rebecca and team, when do you head back to Nome? What is your schedule?
August 15th is when we will arrive in Nome.
And then do you head straight back into the classroom?
Yeah, our school year will start on August 22nd. So I'll head right back and start right into teacher in service and getting my classroom ready.
Wow, exciting. They will be happy to see you and hear all about your summer vacation, your adventures. So with that, we will stop the recording and say thank you